Hello guys, today we'll learn about self-organizing maps, also called as coherent maps. It is named after its author. Before we had traditional neural networks which would handle low-dimensional data, but later on we had high-dimensional data as well as sequential data, and we needed a output which is having relation between two different inputs. So SOMs would handle those kind of inputs. And initially, when the model was implemented, we had an analytical description more on a technical point of view rather than a biological point of view. But later on, learning the results and comparing it with the brain, it had more correlation with the brain. So we adapted this using unsupervised learning algorithm such as k-means clustering, etc. Because it made more sense because brain would usually get unorganized data or raw data. Here we can see the correlation between brain maps and SOMs. The first point tells about training process of SOMs. So in SOMs, we can train the whole model by concentrating on one region at a time. Suppose there are smaller subregions like red, purple, blue, etc. We can first take input data which is related to only this region and then train it and then combine all the results to get the final training model. Similarly, in brain maps, we can understand the whole brain by combining smaller parts of brains like cortex, medulla, etc. The results can be obtained by studying the tumors or malfunctions on these smaller parts. The second the second point is based on fine-tuning models. Fine-tuning the SOMs can be done by changing the weight vector of a neuron temporarily and then using the same input data and checking how much the difference, how much is the loss function, and then using back propagation we can change the weight vectors. And similarly in brain maps, we can induce small lesions. Lesions is basically malfunctions or tumors, temporary tumors, using the electric current and get a good understanding on the region of choice. That is similar to fine tuning basically. The third point would be with respect to semantic analysis. Some regions in the cortex region of the brain are specialized for that, and in SOMs also the application, one of the main applications is semantic analysis. The SOMs constitutes of three major components: neural networks, k-means clustering, and vector quantization. The k-means clustering algorithm is an unsupervised learning algorithm, which means there is no label in the input data. The unorganized data is basically separated into different clusters. The number of clusters is predetermined by the number of centroids. It's based on our choice, and the centroids are also random data points from the raw data. So the algorithm is as follows. Initially, we'll be having data points like this. Suppose there's a random centroid that we have chosen. It's over here and here. We'll choose a third centroid. For each data point, we'll be finding the minimum distance between this. And which one will be having the minimum distance? This one, right? So this data point will be classified into this cluster. This belongs to cluster 1. This will be also in cluster 1. Suppose we choose this data point. This will be associated with this cluster. Suppose this is second one. So this belongs to the second cluster. So like that, we'll have different clusters created. And this is just the first iteration. And in the second iteration, we calculate the centroid again by the k-means clustering centroid calculation formula, and then this process continues until the centroid doesn't change. The final result of the k-means clustering will look something like this. We'll have many different clusters, and the black points which you see is the centroids, the final centroids of the last iteration. Here we can see how the k-means clustering is used in SOMs. We have taken an example over here. This is the concentration of 8 ions in water. Suppose we take a sodium ion concentration and potassium ion concentration. it looks very similar, right? So the top left corner is having a higher response than the uh, other parts, other cells. So finally, the results are combined, and this is the k-means clustering. Here we can see the top left, dark blue color is more responsive to cluster 1. That means certain inputs like sodium ions, potassium, or chlorine are more responsive to cluster 1. Similarly, we can see the bicarbonate ion over here. We can see the right part of the clusters. Sorry, the right part of the SOM is more responsive. So the right cluster is cluster 4, which is responsive to bicarbonates. So similarly, the other ions also. The second component of SOMs is the artificial neural network. The artificial neural networks are a set of neurons arranged in layers, and there are many such layers arranged parallel to each other. And the neurons in the previous layers are connected to the neurons in the next layer. The input data vector is passed to the input layer, and then the weight vectors will calculate the output from each layer. The final output layer will have a certain activation function, a nonlinear activation function such as softmax, ReLU, or sigmoid. Based on the actual output and the expected output, a loss is calculated and it's backpropagated to change the weight vectors. And how the artificial neural network is used in SOMs. There are some key differences that we have to notice. The artificial neural networks have multiple layers of neurons, but the SOMs have only two layers, which is the input layer, which has different features, and the output layer, which is the map. So here we can see this is the input features, and the output layer is just the second layer, and it's just a two-dimensional map. And then the SOMs are unsupervised learning algorithms, but the ANNs are supervised. The third difference would be ANN, the whole network acts as a model. The final vector that we see here, output layer vector, that is one particular model. But in SOMs, each neuron will have certain weight vectors. Suppose the first node over here will have a weight vector of n dimension. So that n dimension will be one model itself. So the ANN is a whole model, is one whole model, but SOM, each neuron is a model. The fourth difference would be the ANN, a neuron is independent on the neighboring neuron. That means this neuron, suppose this one and this one, is not connected with each other. But if you see the Cohen maps, the last layer, you can see the first neuron and the second neuron is connected. Similarly, the second with the jth neuron is connected. So each neuron in the output layer is connected with each other, the neighboring neurons only. Hence, we can store the relative data, that is the input. Suppose there are two inputs, and we can store the sequential input information also in the Cohen maps. The third component of SOM would be vector quantization. In this step, we would reduce the dataset size to a significant level. The word quantization means to reduce the precision from a high precision to a low precision to achieve a lesser response time. The steps taken are as follows. Initially, we divide the whole dataset into different cells, and each cell we would select a representative vector, which is called as the model vector. This model vector is chosen based on the minimum mean quantization error function, in which the minimum distance is found out from every data point to the candidate model vectors, and one of them is chosen. The minimum one is chosen. Here we can see how vector quantization is used in SOMs. Earlier we chose the winner data vector from a set of data vectors within a cell. But here we have a SOM with subregions, and within the subregions we are choosing one of the representative nodes. So using the same formula that we saw earlier, energy formula, so we're gonna choose the data vector from this set. So it's illustrated using this diagram. Here we can see different colors representing different regions in the SOMs. And we can see a white vector, data vector. That would be the representative vector or the model vector. So till now we have learned the three major components of SOMs. Now we'll be learning how these three components contribute to the SOMs, and step by step we'll be learning how the algorithm works. So the first step, we will classify all the nodes or neurons into different clusters using the k-means clustering algorithm that we have seen earlier. The second step will be choosing the best matching unit or the model vector from each cluster, which is using the vector quantization method. And for this, we'll be using the mean quantization, the minimum mean quantization error function. So in the third step, we'll be changing the weight vectors. So we'll be adding a term to the old weight vector value to get the new weight vector of the next epoch. This term will be consisting of the learning rate new, the heuristic value. This heuristic value we'll be seeing later, uh, and then this would be the direction in which the change would happen. So the heuristic value is basically a Gaussian function. This will tell how close the data vector is with the model vector. So, so we can see this Gaussian function right here. This should be a heuristic value. So considering this as the model vector, this is a side view of the SOM. And suppose these are data vectors. We have the heuristic h b of I. So the data vectors which are closer to this will have a higher heuristic value than the data vectors which are farther. The weight vector change that is happening for the nodes that are farther from the model node will get a lesser change, and the ones closer will have a higher change in its weights. So here we can see two different clusters with two different Gaussian functions. The Gaussian sigma value will be initially will be very high, and as the epochs increases, the Gaussian sigma will reduce, and it will become more steeper. Even the learning rate would decrease. This means that we are concentrating on the data points which are closer to the model vector uh, as we go through the training process. So we are doing less change to fine tune the model more. Here we can see the different ways of performing the training process. First one would be a stochastic gradient descent, wherein one input is concentrated at a time and the weight vectors are changed just after the first input is passed, and then each arrow mark over here shows the weight change. So we can see a drastic change in weights in stochastic gradient descent. The second way of performing training is batch gradient descent. Here we pass all the inputs and then the loss function is calculated for the final output. So here we can see that the weight vector is not changed much and it's moving towards the minimum. So this is what is usually done. The last way of performing training is mini batch, in which smaller groups of input data is made and each group is passed and the loss function is calculated for only that particular group, and the weight vectors are changed, and then the next group is passed and so on. So this combines both the stochastic gradient descent and the batch gradient descent properties. Here we can see some applications of SOMs. The first one would be document clustering. That means we can provide any documents as input and we can get the type of the document as the output. Here we'll use different types of documents such as summary documents or land paperwork, etc. as input to this SOMs. And the output of the SOM will look something like this. For class 1 document, suppose it's a summary document of something, then this is the response of the SOM. This will look like a response for class 1 documents. So if the output of the SOM looks like this, then that means that the document is of type class 1 and etc. You can see class 4 as a different type of response. So the second application of SOM would be the Britannica Encyclopedia. So here we can see the final trained model of SOM. Suppose we take a word larva, the related words such as egg, adult, lay, other, etc. would be present in the neighboring nodes of the larva node. So we can think of larva as the model vector and the surrounding vectors as the related words. And the articles also are stored in some nodes. The third application of SOM would be to store 7 million, nearly 7 million patent abstracts. Suppose we store the name of the patent over here in one node. The abstract can be stored in the nearby or the neighboring nodes. Just like what we saw earlier in the encyclopedia, we can store the related words in the neighboring nodes. Here we can store the abstracts of each of the patent IDs in the neighboring nodes. So that's it. Hope this video was informative, and I'm making this video also for my teaching demo assessment at NITK for the course of image processing and computer vision. Thank you.